Hello everybody, welcome to JJZACH Medical. In today's video, we are just going to be briefly looking at something called quorum sensing. Now, if we battle this, is this the right way to fight bacteria? Is it going to be better than using antibiotics? Well, let's just look at a brief idea. So as you can see, there are lots of bacteria in this photo, and in reality, of course, as we know, they are very small compared to a standard human and if you look at it with the naked eye, you couldn't even see the bacteria. Now, then how do these little guys, as small as they are, attack humans? Well, you can think, well, not think, but it's largely because of quorum sensing. Now, here's just an example of quorum sensing, and in this organism, well, not in it, with the help of this bacteria, this organism can see in the night and it can eat whatever. Or any other goal, that's one way it could benefit it. And this is an example of mutualism, because if you have a large number of these bacteria accumulate together, you have a high cell density, and that means you have a high signal density. And then when you have this, you have activated these proteins with the help of these ligands. Ligands just meaning signal molecules. It binds, and then add, and then they go together to the Lux type box, and then you get the bioluminescence, and that's when you get the gene expression. However, when you have a low cell and signal density, so you don't have much of these bacteria, you're not going to activate that Luxar, and then you cannot have that gene expression to create the bioluminescence. So that's not going to be good. Yeah, I mean, you can kind of see here it's going to produce that, and that's going to produce this ligand, and that's not fully coming there like it does here. So that's quorum sensing, really, when bacteria communicate, and they always, accept all these ligands, and then they produce the bioluminescence. When you have a high cell density, you have lots of these ligands, and they'll bind to the Luxar and things like that. Of course, this varies in bacteria. So, of course, there is limitations. As you can see, I mean, with the quorum quenching therapies, this is basically you're trying to stop the quorum sensing between the bacteria. And then there's some problems with this, because you may have a replication rate that could possibly increase. You could have biofilm formation. You could have resistance to antibiotics, another bad thing. You could have increased inflammation resulting from the loss of exoenzymes, DNA repair, metabolism. All these things could be bad. In fact, you can have regenerative processes of the host. Now, is this better than antibiotics? Because the thing with antibiotics, you can kill bacteria, but there's a chance there'll be that one bacteria population or small number of bacteria within that population that is resistant to the bacteria. So you're just basically making them more resistant every time you give them antibiotics. That's a maybe though. So is this the right way? Well, you tell me in the comments. Anyways, here's the credits. Thank you. But ultimately, it's all in God's hands.